My name is Emily, and I'm a PhD student in the group called Microbial Biotechnology and Biorefining, and I have entitled my presentation today, The Golden Microbiome. So first of all, a little bit of background to try to understand the relationship between riboflavin and oxygen, which is more or less all what this is about. My PhD project is a part of a bigger project focusing on bacterial interactions in the gut, focused mostly on electron shuttling compounds, so that could be riboflavin, which is this in this case, which is known to carry electrons both between molecules but also between different bacterial strains in the gut. An example here is a strain called Faecalibacterium duncani, who has the ability to take extracellular riboflavin and reduce it, and together with cysteine, it can use more or less oxygen as the last electron acceptor to produce water. But focusing a little bit more on riboflavin, it's also of a great health benefit. It is known that if you have some decreased levels in the gut, you have a risk, or it's associated with diseases such as IBD and different iron deficient diseases, something with your vision or cancer or migraine. So what we are trying to do is trying to understand the interplay between riboflavin in the gut and different bacterial species and how it affects the host health, but also how it maybe is a response to oxidative stress and how it's influenced in microbial community. So what is interesting about that is that a lot of B2-producing organisms are present in the gut, also in different fermented foods and also in vaginal, so different microbiomes. But there's both producers and non-producers, and these these different producers, non-producers ratios are interesting because it's seen that, for example, with my strain, I didn't highlight it so well, but there is strains of the one I'm working with where it produces riboflavin, but also within species it doesn't. Or it requires riboflavin to grow. So we're trying to understand why they produce or overproduce riboflavin and why this may be a difference in between species. So we started out with using Lactococcus lactis as a modeling strain for lactic acid bacteria, where this have also been seen. And it is known that Lactococcus lactis suffer from heat-induced oxidative stress. And if you supply the media with riboflavin, it can better overcome this stress. So we made a laboratory adaptive evolution where we increased the temperature over time to hope to isolate riboflavin overproducers. And it actually happened. These golden cultures, they contain riboflavin, and it happened already after 2 weeks of evolution. We isolated the strains by using this interplay that is between species. We had Lactococcus that require riboflavin, and then we isolated as overproducers by streaking them on the plane, and then if the background strain grew, we know that they were feeding riboflavin to the surrounding strain. And this is interesting specifically for Lactococcus because it has a lot of genes that encodes flavor protein, and one of these genes are the NADH oxidase. So here FAD helps to cope with the dissolved oxygen in the media or in an environment, and thereby these antioxidative properties can help eliminate the heat-induced oxidative stress that we saw. We confirmed that they grew better with the presence of riboflavin. So if you have media without riboflavin and you put in or ALE strains that overproduce riboflavin, they were able to grow better at higher temperatures. Then we dived a little bit deeper into try to understand the mechanism behind producing riboflavin and what's make them further overproduce. And a little bit interesting was that when we excluded nucleotides, which normally are quite important for growth and also a precursor for the riboflavin biosynthesis, we saw an increase in riboflavin production. But instead we correlated with that when you exclude the nucleotide, you have a slower growth. And we try to mimic the same to the right by focusing on the glycolysis, where if you exchange to maltose instead of glucose, you have an insufficient supply of ATP generated in the glycolysis, and they grow slower. And here the gray bar simulates that they also produced higher riboflavin. So not just the slow growth, but also increasing the biomass will help increase the riboflavin content. So we made a mutant where we reduced the lactate formation that normally is known to inhibit growth when you reach a certain low pH. If you limit the lactic acid production and aerate for producing acetoin, the pH doesn't drop so much, and you again have a higher riboflavin production. Then we scaled it up to 1 liter fermenters, and we were actually able to reach the highest reported titer of riboflavin produced for lactic acid bacteria, which was 64 milligrams per liter. And that was more or less the purpose of this paper that have been accepted in the Journal of Agriculture and Food. But we think it also really helps us try to understand the bacterial interactions and what helps gut microbes maybe cope with oxidative stress. So that's what to come next.